Dear students, you are welcome in this class. And today is 10th April 2023. And this is my last lecture in this course. And we continue with the topic soil-plant relationships. And today we are going to learn the characteristics of plant roots and different methods for studying them. As already told to you that plant roots are very, very important. They are linking the plant to the soil, and the interaction of plants or interaction of whole plant is with the soil is through roots. So roots are the face. Roots are the face of interaction of soil and plant. And definitely roots are very, very important. You already know their functions and how the roots are studied. What are the important parameters that we study in plant roots, like dry matter, root length, and whatsoever parameters we study in plant roots. And you will also see why to study these root-shoot ratio. Root-shoot ratio is also important. So we will discuss all these subjects and describe the types and functions of AM fungi, arbuscular mycorrhiza fungi. They also partly substitute for the functions of the root. They help in the absorption of certain nutrients and water. They increase the absorptive area of the roots. So therefore, AM fungi are very closely linked to the roots. Certain glossary terms, I think you can go through yourself, like hostorial roots. It is just a refresh of your memories, refreshing memories. Primary root, root boss, root cap, you know the end of the root. It is present at the apex of the roots. So please go through all these terminologies. Root hairs are not new to you. And root crown, what is root crown? Where stem and root portion join each other. Where they meet is your root crown. Roots, secondary roots. And you also know there are two kinds of roots. One is fibrous roots, which is mostly common in monocots, and slender branched grow directly from the stem. So please go through the details of fibrous roots. And then tap roots, mostly in dicots, you have tap roots, and single primary root grows deep into soil and remains so. Please go through it. We start here. Already you have seen the soil-plant relations, the scope, what actually we study in soil-plant relation. It is already in your mind. But one new thing here, you can see rootability and rooting zone. This is mostly related to studies of the roots. So root system and rootability, potential rooting zone. So we'll discuss this, this important aspect. Number two, water supply. In soil-plant relation, we study water supply also. So plant available water, you know what is plant available water. Water transport in the soil-plant atmosphere continuum. Water consumption and plant yields. So this subject I'm not going to deal here. You already have a course on this. So means principles and practices of water management, or you have some courses on irrigation or water management. Soil, air and oxygen supply to plant roots. It is also soil plant, part of soil plant relationship. One more thing you can see, soil thermal budget. Soil also influences, or the temperature of the soil is also important in respect to the plant. Soil temperature and plant growth. I'm not going to teach you this one also. Plant nutrient supply. This we have already discussed. Nutrient contents, forms, budgets, availability and supply in soils, and nutrient fertilization. So whole course is devoted for this, that is soil fertility and nutrient management. Soil colloid, soil organic matter, soil organism vis-a-vis plant growth. I've already given you yesterday 2 presentations on soil organic matter. Just browse those slides. It is not part of your syllabus, of course, but you would learn something about soil organic matter. Now we start the plant root, the actual topic of today's. So let us see general information about plant roots. So most root biomass in annual crops, this we are talking about annual crops, not the perennial shrubs or bushes or trees. So most root biomass is in 0 to 20 centimeter soil depth. Root system in cereals, it is fibrous. In legumes, it is taproot. Cereals are more vigorous than legumes. So overall, the cereal root system is more vigorous than the legume. This is with respect to total biomass. Root contributes 10 to 20% of total plant weight. This is important. You can underline this. These are very general values. Root contributes 10 to 20% of the total plant weight. Deeper and vigorous, if you have very deep and vigorous roots, so absorption of water from deeper soil layer is possible. Root length and weight have positive effect on grain yield. So you can see that you can have a response curve fitting. You can take root length of different plants or root length of different varieties, whatsoever you like, and or you can take root weight and see the association with the grain yield. So it was found out to be positive. Root dry weight, it has very high correlation with grain yield. So that means generally higher the root weight, higher is the grain yield. C4 plants have more vigorous root system than C3 plants. You can see the taproot system on the left side, and right side you have fibrous root system, which is also kind of adventitious roots. Now, if you want to make the root studies, and there are two important methods, greenhouse or container methods or field methods. In greenhouse or container method, you can grow plants in containers, and those containers can be kept in greenhouse, or you can have some glasses, container of glasses. Variety of options are available, and you grow the plant in them and study the root. Similarly, you can study roots directly in the field also, and some real-time measurements are available in the field, and some you can do by scanning or scanner. Lot of softwares and scanners are available. Now, in container methods, options could be rhizotron, mini rhizotron, rhizometer, hydraulic conductance flow meter. So these are the options for container methods if you're following. And digital imaging is also there. You can take root samples, and image analyzing computers are available. WinRhizo is a software, Root Reader, NMR, and X-ray CT. So number of options are available in this case. The main thing is the most common method that is used by researchers in India or by students is simple. Take the sample with core sampler, and then you can have a monolith also. Core sampler takes the sample in core. Monolith means you can cut the soil in a square or rectangular shape. It depends upon your choice how much area you want to sample. So monolith is just getting the roots with soil. Wash after sampling the soil, you wash the soil and clean the roots. Use root scanner. Then after removing the moisture from the roots, you can use the root scanner to scan. This root scanner will give you a lot of data related to root. It will give you root length, root diameter, or total root length. Variety of observations or parameters will be given by the root scanner. So these days, you have variety of scanners. This computer or IT technology has entered into laboratories very much. So overall, what you get from these root scanners, root length, root number, dry weight. Dry weight you cannot get from the scanner. You have to oven dry it. The surface area you can get from scanner, root volume you can get, root diameter you can get. So these parameters are very, very important. Suppose root length. Generally speaking, if you have more root length, then it will cover more portion of the soil. So similarly, root number, dry weight. Generally, more dry weight means more root biomass, and comparatively, the roots can explore larger volume of the soil. But many times this dry weight can be misleading because we do not know the diameter of the root. Finer the diameter of the root, better is the uptake of nutrients and water. Surface area of the root is also important, and root volume and root diameter. So different parameters are studied for root. Just they are seeing the growth of the roots. More growth generally means that it is covering the larger proportion of the soil. For then you will have more nutrient availability and uptake and water uptake. Now, plant roots and soil relations, the density and distribution of roots affect the amount of nutrients and water that roots extract. So this is influenced. The uptake of nutrient and water is influenced by roots. Root distribution and density are a function of kind of plant and nature of the root environment. So both are important. Genetics is also important and environment is also important. Sometimes environment may not be in favor of root development, and sometimes it may be in favor of root development. So here environment can also include the soil type, the kind of soil, whether it has rocks beneath the plow layer or it has some restricted environment. There may be some restriction for the root growth in the soil. And also the water relations are also important for root growth. In aerobic crops or aerobic plants, if you have too much of water, root growth will be adversely affected. So root systems of plants and rootability means the extent to which root explore the soil. The area that is being covered by the soil is rootability. Roots at maturity, remember this statement, roots at maturity are approximately 10% of the entire mass of the cereal plants. Means the 10% weight, the dry weight of the whole plant is root. In corn, wheat, and oats. So oat roots, for example, different workers have reported different data. 13% of the total plant weight in oat. Trees common range of 15 to 25% of entire biomass. In trees is root. Root supplies the main material for organic matter transformation in soil. The root system are two kind of root systems are there. Students can remember this. Two kind of root system. One is coarse root system having diameter more than 2 millimeter. That will be your coarser root. And fine roots means less than 2 millimeter. These are fine roots. Root hairs are also there. Thickness of 5 to 20 micrometer. This is the thickness or diameter of the root hairs. Length up to 1000 micrometer for root hair and remain functional for a few days. They do not survive long. The new root hair replace the older one. Coarse and fine roots penetrate into coarse pores of the soil. So both kind of roots, coarse as well as fine, because their diameter is around 2 micrometer or somewhere around it. So they can enter the coarse pores. Both can enter the coarse pores, which are more than 10 micrometer diameter. Root hairs penetrate into a large portion of mesospores. So 2 to 10 micrometer are mesospores. This mesopores, this enables the uptake of water and nutrients. In the fine pore range, that is less than 0.2 micrometer, these substances only reach roots through slow diffusion along concentration gradient in soil solution. Oxygen supplied to roots through large coarse pores, that is more than 50 micrometer. RLD frequently reaches several meter per cubic decimeter. RLD is root length density. Now, root distribution of different crops. At least half root biomass in the upper 20 centimeter of soil for all crops. So you have already seen that majority of the roots are concentrated in top 20 centimeter layer, particularly in annual crops like oat, barley, lentil, soybean, canola, fescue, and the shallowest D50. D50 is less than 13 centimeter. This D50 means D50 is the depth of soil where up to which the 50% roots are there. The depth of the soil where up to 50% roots by weight are there, or biomass of the root. Suppose it is less than 13 centimeter here. So that means that 50% roots are there in the 13 centimeter upper layer. So that is D50. Similarly, you can have D60, D70, and so on, depth. So D50 with 67 to 76% of the roots in the upper 30 centimeter. Wheat, pea, chickpea, and alfalfa with 61 to 68% of the roots in the uppermost 30 centimeter soil profile showed deeper D50, more than 15 centimeters. This I am giving you just to have idea that how much biomass is there in the roots and what is the depth of roots. Alfalfa have deepest rooting profile with 95% of the roots in 136 centimeter. You can see the legume crops generally have deeper root system, a maximum rooting depth of 177 centimeter. Wheat, barley, soybean, 95% of the roots occur in top 100 to 138 centimeter and Dmax of 146 to 172 centimeter. The maximum depth to which roots have been noticed. Or pea, chickpea, 95% of the roots in top 64 to 85 centimeter and Dmax is 78 to 110 centimeter. Now see this fact with the time, how the root growth occurs. So you can see here, days after planting, this is maize crop or corn, and this is your emergence. So you can see effective root depth. One is effective root depth, the thicker one, and other is maximum root depth. So just see the effective root depth. So you can see that it is following a sigmoid kind of curve in the beginning, and then it becomes static. So it is increasing at a faster rate initially, then slower rate, and then it stops increasing. So you can see that after the increase in root growth or depth, you can say corn root depth, increase in depth or size of the root was only up to reproductive stage, tasseling and silking, and beyond which there is no change in root length or root depth. So you can say that the root growth is stopped after reproductive stage. Then the materials which were getting transferred to the root from the photosynthesis will now get to the sink. So now root will not be given preference by the plant. See this case also, field observe plant parameters for the maize crop. Root depth, plant height, leaf area index. So you can see they are particularly root depth and plant height. They increase only up to the reproductive phase at about 60-65 days. And then they start falling. Even the root depth or root growth start falling because there may be decomposition of dead roots or detachment of some certain root parts. So you can see maximum root growth is up to flowering or up to reproductive stage, and then there may be slight decrease in the root depth or root growth. Similar is the case of Indian mustard. You can see maximum root growth or root depth is up to this reproductive stage. Now you can see the rooting pattern of different crops, pea, corn, potato. This is how the roots are distributed. Wheat, winter wheat, this is spring wheat, and then alfalfa. You can see very deep, very deep. Now you see the root distribution in case of rice. Even within a crop or within a species, you can have different distribution of the roots. Like here, there are different varieties, 4 different varieties of rice, and the root distribution is different. See the first case, it is Jaya variety. Here you can see the root characteristic is shallow and spreading. The rooting depth is less. The rooting depth is less. They are shallow, but they are spreading in nature. You see, they are spreading in nature. So 86% of the roots are there in 16 centimeter soil depth. You can see here. So you can say majority of the roots in rice are in within 20 centimeter of the depth. See the another case here, the deep spreading. The roots are going deep also. You can see they are going deep up to 24 centimeter or more, and they are also spreading. They are also spreading. See the third case, where you have shallow and compact. The root depth is shallow. 90% roots are in 16 centimeter depth, and they are not spreading. They are very compact, covering the small fraction of the soil. And then see the fourth one in Padma variety, where you have deep compact. Deep, the root growth is deep, you can say going deeper up to 32 centimeter, but it is compact. Thus lateral, the side growth is less, they are compact. So now you can see 4 different kind of rooting behavior in rice: shallow spreading, deep spreading, shallow compact, and deep compact. So in your view, which kind of rooting system would be better out of these 4? Yeah, Satyapal, in your view, which system would be better for nutrient
Sir,
uptake? Second.
Sir, second.
Yeah, this one. Why? Deep spreading, why?
Sir, absorb more nutrient from these roots due to
Exactly.
higher surface area.
Yeah. Yeah, very good. Actually, in this case, deep spreading, the spread is more. Spread is more. That means it is coming in contact with larger volume of soil. It is exploring more volume of soil, so it will get more of water, more of nutrients. In other cases, its spread is low. See, in another case, here also you can see this is root distribution pattern in 2 species of jute, Capsularis and Olitorius. You can see in one case it is very deep and compact. In other case, it is shallower and spreading, shallower and spreading. So in these 2 cases, which one would be better? In these 2 cases, you see they are going up to 50 centimeter. You can see in one case. In second case, they are going up to 80 centimeter. Which should be ideal one here? I think it is Capsularis. You can see the 40 centimeter depth is enough, I think, for these kind of crops. But you see they are spreading more towards side. So that means this layer of the soil is rich in nutrients also. So plants will get more nutrients here. But if you go to the subsoil, it may not contain sufficient nutrients. So this way, it will extract more of nutrients. Now, potential rooting zone of soil is depth of potentially rootable soil zone where vertical root growth is not restricted. So that is your depth of potentially rootable soil zone where vertical root growth is not restricted. Means plant can go as deep as it wants by mechanical or physiological boundary. There is no any restriction of mechanical nature or physiological nature. So mechanical nature or boundaries are compacted layer. There can be soil compaction, which will restrict the growth, and hardened subsoil horizons can be there, sea horizon, over compacted calcareous glacial till, hard rock or stone layers. So there can be mechanical boundaries which can reduce the potentially rootable zone. And there can be physiological boundaries like water-saturated subsoil horizons with oxygen deficiency, strongly acidified subsoils with aluminum toxicity. So these are physiological boundaries because of which the root growth can be restricted. The rooting zone in soil, there can be 2 or 3 kind of rooting zones in soil: shallow, intermediate, or deep rooting zone. Just now you have seen. In addition to vertical rootability for continuous root growth, the horizontal rootability of soil is decisive or important for the rooting intensity of soil horizons. Unfavorable structural forms such as porous, polyhedral, and prismatic structure in clay-rich soils only have low core spore fractions and therefore impaired horizontal root development. Now we go to the root shoot ratio. So the root weight divided by the shoot weight. Here it is dry weight of the root and dry weight of the shoot. Or you can say dry biomass of the root divided by dry biomass of the shoot. So why to study the root shoot ratio? It is mostly studied by the plant physiologists to know the distribution of photosynthetic product in the roots and shoot. What proportion of photosynthetes is migrated to the root and to the shoot. To compare below ground biomass, including root exudates, as a function of shoot biomass, including grain. High root to shoot ratio used in breeding crop genotypes for broad drought tolerance. So this knowledge of root shoot ratio can also help in the breeding varieties for drought tolerance. So these are the purpose of studying the root shoot ratio. Remember, it is always studied as root to shoot ratio. Or partitioning of dry matter in roots relative to shoots is high during the seedling stages of growth and steadily declines throughout the development. The root shoot ratio decreases with plant age. This is important. This point you can underline. Root shoot ratio decreases with plant age, indicating that the major part of the photosynthesis is going to the shoot. During early vegetative growth, up to 50% of the assimilated carbon translocated to roots, of which 50% consumed in respiration or lost from the plant as rhizodeposition. After anthesis, the amount of assimilates translocated to the root system is very small. So you can see after anthesis means when the reproductive stage has started, the translocation to the root system is very small and the net size of the root system hardly increases and even declines after anthesis. So factors like temperature, solar radiation, moisture, fertility, soil texture, structure influence the root shoot ratio. Crop species and genotypes also influence the root shoot ratio. So you can see Huggins and Foucault's root shoot ratio 0.25% for corn. For wheat, it can be 0.28. Again, some other worker has reported 0.21, 0.23. It is giving you general idea, or you can say 1/4 or 0.25. They also reported RS ratio 0.48 for wheat, 0.35 for corn. So you can see these ratios are varying. Depending upon the plant type, the stage of the plant is also important. At different stages or varieties will have different root to shoot ratio. So a lot of variability exists. You cannot generalize the values. So you can see in this case how they are varying the root shoot ratio. Root to shoot ratio of upland rice, corn, wheat, soybean, and dry bean during growth cycle. This reference is from Fagheria and co-workers. Here I don't have here. I just taken it from his book. So he has written a book on roots, plant roots. So I taken from that book of Fagheria, N.K. Fagheria. So now you can see the plant age in days, 21 days. What happens? You see very small ratio of root shoot. So because initially seminal kind of roots are there here. So 0.04 in corn, 0.25, and you can see this. At 40 days, it increases. It increases. In wheat, it decreases. So you can go through this data and see and observe the root shoot ratio. Root length and root density are important. They determine the root surface area. Root surface area determines the water and nutrient uptake potential of roots. Root acquisition of nutrient and water is based more on root length and root surface area than mass, since high root length density is linked to a short travel distance of water and solutes. So you can see how this root length is calculated. Newman has given a formula because in those days, root length used to be measured by graph paper. So you will have a big size graph paper and you have roots. Just spread the roots, cut all the roots into pieces and spread them on the graph paper and then count the and measure the root length. So Newman developed a theory that root length can be estimated as R is equal to pi an upon
The
2.
slide is not visible.
Slide is not visible. Is it moving?
No, sir.
Why didn't you say it? Anyway, so we can join to the new link because it is only 5 minutes left. So I can stop share and let me check it again. So you can join the new link and I can stop the recording also. So, you can see Newman has given this procedure. Here R is the total length of the roots in centimeter. You can report in centimeter, in millimeter, any unit you like. N is the number of intercepts between the root and the straight lines on a grid. This is on graph paper. A is the area of a square or rectangle of the grid. H is the total length of the straight lines. But now these days, nobody is using this formula. Most of the people have now equipments and cameras, machines, scanner to know the root length. It is very tedious. It takes a lot of time to do this exercise and measure the root length. You can see this maximum root length of 5 crop species during growth cycle, the different crops. So you can see the root length, just to have some idea that in upland rice, for example, the root length varies from 20 to 25 centimeters, just to have some idea. So initially it is low, then it starts increasing, and finally at maturity, it is reduced. So 9, 19, 24, 24, 26. So this is the pattern. Again, from Fagaria, I have taken this table. Corn also, you can see it is increasing, and then it starts falling. And in the end, it is more, I don't know. So these are the trend and range. At least you can see that in maize, the root length is 40 centimeter. Here, root length means length of all the roots, whether it is whether it is, you see in rooting, you get branching also. So every root is broken down and the total root is measured. So in wheat also, you can say in wheat, total root length is around, you can say 20 centimeter, you can take it. In soybean, it is about 25 to 30 centimeter dry bean. So this is just to give you idea, how much is the total root length. Root length per unit volume. One more measurement is there, which is indirectly measured from the root length. It is root length density, RLD. Root length per unit volume of soil. RLD is root length per unit volume of soil. So you can see root length density is in centimeter per cubic centimeter soil. So total root length in centimeter divided by soil volume where roots have been collected in centimeter square. A specific root length is the ratio of length of mass, that is the SRL. So you can see root length in centimeter divided by root dry weight in gram. Root dry weight is very easy. You can take the sample of the roots and put it at 105 degrees centigrade for about 10 to 20 hours. And then you will get the dry weight. Or otherwise, some people like to take the dry weight at 60 to 75 degrees centigrade temperature. But here the time will be more. You need to have about 48 hours time. But this is quicker. So it depends upon what you want to do. If you are going for nutrient analysis, then better is to keep the lower temperature. Is it changed? The slide is changed. Root development relative to plant growth stage. Now, we go to mycorrhiza, role of mycorrhiza. You can see how it is related to root. So you can see in this case, it is ectomycorrhiza. Mycorrhiza, you can say it is forming a very thick structure outside the root. So this is ectomycorrhiza. You can see here, ectomycorrhiza. sometimes it can enter, the hypha can enter or mycelium can enter the intercellular spaces in ectomycorrhiza. So as you know that it is the association or relationship of roots or roots of higher plants with certain species of fungi. So actually roots co-evolved with fungal partners to form mycorrhizas, fungus roots. In an undisturbed forest, almost all trees, irrespective of their taxonomic affinities, are interconnected by a diverse population of mycorrhiza. So they are very common in nature. All the trees in any forest area are linked to each other through the network of mycorrhiza. The roots, they are connected. Roots of two different trees or plant can be connected by mycorrhiza. So you can see this case here. This paper was published in Nature in 1997. So you can see there are two plants, and their roots are connected by mycorrhiza. Certain mycorrhiza is connecting the roots of two plants. So roots of up to 90% of the trees in forest can be colonized by many fungal species, mostly ectomycorrhiza. For trees, it is ectomycorrhiza linking two conifer. Here, they are temperate species, provide channels for the transfer of nutrients. So therefore, one plant can give nutrient to another plant and vice versa. It can be bidirectional. It is not that only one plant will give nutrients to this one of carbon. It can be both ways. So considerable carbon can be transferred in either direction between trees through these networks. It depends upon the requirement. And plants are also literate. They are also very intelligent people, you know. So if one is having excess carbon, he can share with the other partner. And if other partner is having excess nitrogen, it will share with this partner. So it is kind of mutual relationship. So a symbiotic association between a fungus and roots of a vascular plant. Important roles in soil biology and chemistry. Fungus colonizes the host plant's root tissues either intracellularly. Intracellularly means within the cell. The hypha will enter within the cell, not between the cells. So intracellularly, when it enters intracellularly, then it is endomycorrhiza. But instead of endomycorrhiza, now people call it by the name AMF, arbuscular mycorrhiza fungi, or in brief, AM fungi. So before, the name was VAM, vesicular arbuscular mycorrhiza. So this vesicular arbuscular mycorrhiza name has been deleted. Now it is not used in the scientific literature. So VAM has been replaced by AMF, arbuscular mycorrhiza fungi, because all the arbuscular mycorrhiza fungi were not making the vesicles. They were not making the vesicle. So therefore the VAM, vesicular word, was dropped. So they can be intracellularly or extracellularly. Extracellularly means in the intercellular spaces, ectomycorrhiza. Now you see ectomycorrhiza, hyphae surround but do not penetrate the root cells. It may surround the roots, but it may not penetrate the root cell always. But some can penetrate the root cell, but it is in the intercellular spaces. Trees in temperate region, gymnosperm and angiosperm, they mostly have ectomycorrhiza. Trees become more resistant to cold, dry conditions. You see, so it is giving some kind of abiotic stress tolerance to the plant. A thick mantle of hypha covers the root surface, and mycelium extends from the mantle into the soil. So this mantle is actually thickness. This is called as a mantle. This thickness. So mycelium extends from the mantle into the soil. Now you see in ectomycorrhiza, inside the root, you can say that it is inside the root, but in intercellular spaces, intercellular hyphae does not enter the cells. Ectomycorrhiza outside the root, it is always outside the root. Two kinds of ectomycorrhiza. thick layer of hypha around root, fungal sheath, lateral roots become stunted. So it remains just outside the surface. Hyphae, mass about equal to root mass, forms extensive network of hyphae, even connecting different plants. It connects different plants. Now we come to the main target of this. This is your arbuscular mycorrhiza fungi, AM fungi. Most common, vast taxonomic range of plants, both herbaceous and woody species. Aseptate hyphae is aseptate, enter the root cortical cells and form characteristics arbuscules. Therefore, name AM fungi. Arbuscules are the intracellular tree-like branching structure that the fungi form within root cortical cells. So these arbuscules are very, very important. Arbuscules are important because essential locations for exchange of carbon compounds from the plant for mineral nutrients, particularly phosphorus from the fungus. So our arbuscules are the site of exchange of material between host plant and the fungus component. So here, for example, if plant for mineral nutrients, particularly phosphorus from the fungus. So this phosphorus is supplied from the fungus. And where from this phosphorus will come? It will come from the soil, because fungi is connected to the soil also. It is there on the roots and also connected to the soil. The hypha of the fungi can go deep into the soil, and it can go to the spaces where root hair cannot enter. It can enter even very, very micro pores where your root hair cannot enter, and it can extract phosphorus from there. Or not just phosphorus. This mycorrhiza has been known to extract or to uptake the zinc also. Many other nutrients are being absorbed by mycorrhiza. It is not just phosphorus, but mainly it is phosphorus. And also it solubilizes the phosphorus in soil. It releases some organic materials, and that solubilize the fixed phosphorus, which is in inorganic form. So this is the advantage of this mycorrhizal fungi. Now you can see the structures here, arbuscules. So see, this is hypha outside the surface of the root. These are different parts of the root. And you can see some AM fungi can also make the vesicles. These are the structures for spores, I think. And they are inside the cell also and outside the cell also. And you see, this is important. This is your arbuscule. So these are the structure, branched structure, which are site of the exchange of materials between host and fungus. And you can see the very close electron or very close picture of the arbuscule. It is, you can see branched structure. More clarity about this arbuscule, how it looks. Now, functions: increase absorptive area. The important functions of AM fungi: increase absorptive area of roots, 10 to 1,000 times more absorptive area, increase uptake of soil resource, that is water and nutrients, absorb and transfer almost all the 16 macro and micronutrients necessary for plant growth, but predominantly it is phosphorus, iron. So release tightly bound soil nutrients like phosphorus and iron also. Protection of host plant from abiotic and biotic stresses. This is also important. Act as a conduit for communication between linked plant. So communication, it is just like a wire between two plants. And here you can see this picture. You try to follow, try to understand the whole picture. But here I wish to show you the role of mycorrhiza. Just see this legume plant. Legume plant is there, it is fixing nitrogen. And this is non-legume plant, cereal. When cereal and legume are grown together, what happens? There may be direct nitrogen transfer by mycelial network. So this host can give nitrogen to the associated cereal plant, you can see. So this has been conclusively proved now. Role of mycorrhiza is very, very important. So whenever you have intercropping system, so your cereal component is benefited more by the legume component. Please see or try to understand this cycle. This is very excellent, giving a lot of information. And at this stage, you should be able to interpret the meaning of a picture, meaning of a figure. Try to understand yourself. And most mycorrhizal species exhibit low host specificity, allowing for a connection between dissimilar plants to be common. Means they are not very host specific. Most of them are very generic kind of things. Presence of AMF can increase the transfer of symbiotically fixed end from legume to non-legume. So these are the evidences which show that nitrogen can be transferred. Current transfer of nitrogen is there. White clover to perennial ryegrass can transfer, barseem to maize, pea to barley, soybean to maize. There are many examples, mung bean to rice. So studies have been made that confirm that during the growth period, legume can transfer nitrogen to the cereal component with the help of mycorrhiza. Direct nitrogen transfer through mycorrhiza in legumes, non-legume association can be bidirectional. Sometimes it can be bidirectional also. Suppose your legume is suffering from nitrogen deficiency and cereal has sufficient nitrogen, then it can be contributed by cereal to legume also. So therefore, it is bidirectional also. And it is not for nitrogen. Other materials can also be transferred. So there are very good evidences, and I hope that you did not enjoy this lecture much, but anyhow, I just finished this lecture. I have some cough and cold today. So if you have any question, you can ask question.